Now, the coming to the anther culture here, the first point, first step is to be done here that first we need to go for identification of the plants from which the haploid culture has to be established. Nothing but, so if you're working with a particular plant, you need to identify the healthy plant. Okay, now from that you need to go for selection of immature flower buds. Nothing but immature stands for here on a general note. Immature stands for unopened flower bud. So usually what happens as and when there is a progress in the development of the flower, so we are going to see the gynoecium and the androecium are going to be developed. So in the androecium, we are going to see formation of microspore mother cell to the microspore mature, microspore tetrads. These microspore tetrads can also be said as the pollen grains. When the pollen grains have got matured enough, they may start dispersing from the anther. Okay, now once the anther has started dispersing those pollen grains, it may happen the self pollination or the cross pollination. Okay, now, so the point is that we need to select the flower buds before opening of the flower, or the immature flower has to be selected, and that after the selection of that immature flower we need to go for surface sterilization. So what do you mean by surface sterilization here? So since the flower which has been grown in natural condition, there is a possibility of some contaminants. It may be anything other than the pollen grain, other than the anther, other than the flower, anything which is there can be said as a contaminant. So usual in the flower which may be having the contaminant just like microbes, bacteria may be there, or a fungi may be there, or some other microorganisms which has to be removed, because when we are talking about in vitro culturing of anything, the main principle or the main step to be employed is that removal of surface adherent microcontaminants. So that has to be carried out by surface sterilization. That usually the surface sterilization of the flower buds can be done to avoid fungi, spores, or a mycelium, or a bacterium, or a bacterial colonies, or other spores. Okay, now, so you can employ any type of the surface sterilization. So you can use mild to high concentration of different sterilizing agents. Okay, now, based on the capacity of the flower and the type of a flower. Okay, and based on that, because excess amount, the same concentration of the sterilants cannot be used for surface sterilizing in all the type of a flower, because there are some flowers which will be very delicate. There are some flowers which can hold the external chemical pressure which have been exhibited by the different sterilizing agents. Okay, and based on the type and the capacity of the flower, we can employ surface sterilization. So usually surface sterilization can be done by a low concentration of the mercury chloride, and we can use sometimes the antifungal agents. We can use sometimes a low concentration of ethyl alcohol. So these are majorly used as surface sterilizing agents to remove the adherent or surface contaminants. Okay, now, and after the surface sterilization of the flower buds, we need to go for removal of anthers. This removal of anthers from the flower has to be done in aseptically or aseptical chambers. Nothing but the person one who is working with the selection of the flower buds after surface sterilization, the particular flower has to be taken to the laminar airflow or a biosafety cabinet where there should not be any contamination, because we are working with the in vitro culture. Once the anther has been isolated from the flower bud, that has to be inoculated on a nutrient medium. If suppose randomly, if you conduct the isolation of the or a removal of the anther from the flower bud in a natural or in an open area, then there is a chances or a possibility of the environmental or a laboratory microorganisms may come and cause a lot of contamination onto the anther. So during or when we are culturing those on a nutrient medium, so the removal of the anther from the immature flower bed has to be done. Along with that, aseptically has to be carried out. After once the anther has been removed from the flower, there should not be any filament which has got attached to the anther. Nothing but we are supposed to inoculate only the anther. Precisely, the culturing of anther should be done without the involvement of any of the type of cells or tissue. So when it is a stamen, we are supposed to remove the filament. Only the anther part has to be taken, and that has to be inoculated on a nutrient medium. The point is that so directly we can go for inoculation of anther, but the chance or a rate of success or a frequency of success is very less if we go for culturing of anthers randomly without studying at what stage, what is the meiotic stage of anther required for culturing. So to get better results, we need to identify a proper stage. Okay, now. So for that, since anthers are the reproductive structure that we are going to see, the usually we are going to see the reductional cell division which is going to happen. That's what we call them as a meiosis. So the meiotic stages has to be carried out by using the acetocarmine a nuclear stain. So once the anther has been removed from the flower bud, that particular anther should be detached from the filament. Only the anther has to be taken for studying at what stage the anther is that. So you may check meiosis one or meiosis two. If it is in meiosis one, what are the different stages? Prophase one, metaphase one, anaphase one, and the telophase one. Again in prophase one, we have again five divisions, the stages. What are those? Can you tell me those five different stages of the prophase one? Okay.
Leptotin,
In the prophase one, we have again five different stages. Leptotin, packaging.
zygote,
Yes.
diploin,
Okay. Okay. So in prophase one, we are going to see the five different stages. One is leptotin, zygotin, packaging, diplotin, and diakinesis. Okay, now, so must be very quick when you ask the questions. Okay, now. Only waiting for something. I don't know in future if I ask the name of a student, you will wait for sometimes to say whether you are confident about your name or not. Okay, okay, just leave it all those things. So we have to study the different stages. At what stage can be a beneficial for the anther culture. Nothing but it should produce a healthy, viable haploid plant. So instead of waiting for a longer period, okay, na? So and again this the selection at appropriate stage-wise selection of anther again varies from the species to species and the type of a plant to plant. So all the anthers cannot give rise to a good haploid viable plant when you take at a zygote stage or at a metaphase one stage. Okay, now so a different type of a plant can be can produce a haploid viable plant as a different stages. Okay, now. And after identifying an appropriate stage, the anthers are supposed to be gently separated. Nothing but there should be agglomeration. Nothing but these anthers which are got what do you mean compactly arranged. Okay, na? So that has to be gently separated, and only the anthers which have got intactly arranged. Nothing but there should not be any damage in the structure of the anther. Okay, na? Anther, all the parts of the anther should be very intact. That particular anther has to be taken and that has to be inoculated on the nutrient medium. So during the course of isolation, selection, and when we were studying the stages of the anther, if there is any injury or a mechanical damage or a physical damage onto the anther, those anthers should be avoided. Because what happens, in such cases, only that particular anther and this anther, uh, the anther, nothing but it contains both the pollen as well as all the parts of anther walls. It may be having the epidermis, endodermis, and the even the tapetum and even the central nutrition central cavity which is have holding the pollen grains or microspores, everything will be there. Okay, so if there is any such damage, physical damage or any just like shearing, break in the anthers, those should be avoided for taking into the nutrient medium. So what happens? Such usually leads to the formation of callus. Okay, these callus may go for production of either because of the presence of Okay, we'll tell you the limitations in the next coming slide. So usually putting those injured anthers may lead to the formation of callus, greater amount of the callus. Okay, now, so and next coming to this, after once the inoculation has been done, once you are selected a healthy, good, and a viable anther, when you inoculate it on a nutrient medium, those cultures has to be maintained in a culture room with appropriate light and dark conditions. Okay, and you are supposed to provide appropriate temperature too, because with varying the temperature or variation in the light and dark conditions can lead to the variation in the physiological conditions of the anther cultures and leads to the different results. Okay, so usually for anther culture, light and dark conditions should be provided with 12 hours of light and 18 hours of dark. Pardon? Are you getting my point?
Yes, sir.
Okay, 12 hours of light to 18 hours of dark can be given, or you can even reverse it, 6 hours of light and 12 hours of dark condition. So again, the condition, the employing the or setting the light and dark condition in the culture room again depends, but depends on the type of a plant. But the thing is that general requirement of a light and dark condition should be subsequently maintained per day, or after the completion of 12 hours of light, the automatically it has to switch on to the dark condition for 18 hours. After 18 hours of a dark condition, it has to be brought to the 12 hours of light, or we can maintain it 6 hours of light and 12 hours of dark condition. So it should run in a cycle. Once light condition, then it has to, after completion of that particular period, that has to undergo the dark condition, and the temperature should be kept at 28 degrees Celsius. And usually a low light intensity should be maintained. Usually 3600 to 4000 lux light intensity will be maintained for the conventional plant tissue culture laboratory conditions, and where in the anther culture it may require less than 2000 or 1500 of light intensity. Okay, na? And after once all the appropriate conditions are provided for the anther, then the anther starts proliferating because of the potency, because of the capacity to divide. The anthers start proliferating, and these anthers initially may form the callus or directly the embryos, or they may form the callus. These callus later on forms the embryos, and these embryos we call them as somatic embryos. Okay, now, since here we are using it anthers, because of that I am not using the word as somatic, because the somatic, the word should be utilized or can be used correctly for the somatic cells or embryos which have been derived from the somatic cells. Okay, since these embryos which have been derived from the callus, those callus are obtained from the proliferation of the anthers, henceforth the word which has been mentioned as only the embryo. Okay, and again these embryos which will again undergoes the globular stage, heart stage, and the torpedo stage, later on the cotyledonary stage. Okay, now after once the embryo which is fulfills all the requirements for its growth and developments, finally we are going to see the haploid plant. Nothing but, since the anther which will be usually contains the uni or n set of chromosome or a haploid set of chromosomes, the plant cell which will go which are going to regenerate into a complete plant, each and every cells comprising only the haploid or gametophytic conditions of the chromosomes. Okay, now this can be a haploid plant. Okay, now so this is how we have to undergo the anther culture, and hope so that there is no doubts now. We'll move to the next condition, that is what the pollen culture. And in the pollen culture, now we have completed the anther culture, that then we have to study even the pollen culture, because these pollen can also be utilized for the production of haploid plant. Since what we have done in the first step, that is the selection and identification of the appropriate plant, similarly here also you have to identify and select based on the person one who is working with that particular plant, on which plant the plant breeder or a person one who is wants to produce the haploid plant. Accordingly he has to select immature flower bud, As I already told you that why the immature flower bud has to be selected, because the pollen should be immature, it should not be a matured one. So once the maturation of the pollen occurs, then that particular pollen may be shed off and they may start their natural phenomenon. That is what we call them as pollination. It may be the self or it may be the cross pollination. Okay, now so before the commencement of the maturation stage in the flower, the immature flower bud has to be selected. That's what a flower bud, flower in a bud condition, where the immature pollen grain should be there. Okay, now, and those after that, you need to go for extraction or isolation of the pollen grains. Here we are not supposed to be stick on to the anthers. Okay, now only the pollen grains from the anther has to be selected, and those pollen grains has to be taken in a suspension, nothing but in a liquid form, usually containing the pollen retaining medium. So because the pollen grains, they may look dusty like particles, but for their viability, for their longer or for further studies, those needs to be maintained in a suspension, which suspension stands for usually contains a carbon source liquid medium. So what happens, these pollen grains, when we put it in a suspension, there you need to go for removal of all the anther tissue debris. Nothing but what are the anther walls, epidermis, tapetum, some of the debris, tissues which should be removed, only the pure pollen culture or suspension to be retained. And after that, a pure pollen suspension has to be selected, and only viable and the larger one, the larger and the viable pollen has to be selected, where usually the smaller one where dormant pollen grains which will not give a better result. Okay, now usually smaller pollen grains which are having very less chance or a frequency or a rate of production of haploid plants, henceforth a large and pollen grains. So among the different pollen grains, you need to identify which or what are the different we need to identify the size of the pollen grains which are showing a better or a greater size. Those has to be selected, and those which must be viable, nothing but there should not be any damage, there should not be dormant, only viable and the larger pollen grains has to be selected, and those needs to be filtered. Okay, so after the filtration, the collected pollen grains has to be washed, and after the washing, those collected, the pure, viable, and the larger pollen grains has to be collected, and those suspension which comprising the viable, larger, filtered, washed on the collected pollen grains, that has to be inoculated directly on the liquid medium or the solid nutrient medium. Okay, now just here I have inoculation of pollen on broth of solid nutrient medium material. Just make it as R. Okay, this is not off, this is R. So you can inoculate the pollen suspension directly on broth. Broth stands for a medium devoid of agar, a liquid medium, or solid medium, nothing but a medium which comprising all the micro and micronutrients which are similar to that of a liquid medium. The difference is that the solid medium contains a solidifying or a gellifying agent. I think everyone know what is that, what is the solidifying or gellifying agent. So once the liquid or solid nutrient medium has been prepared, and when we have inoculated all those on the pollen suspension on a nutrient medium, then we need to culture it, incubate those pollen suspension which have been inoculated in nutrient medium for different time periods, which have been the same thing what we have done for the anther culture. Again, maintaining for about 12 to 18 hours of light and dark conditions respectively, or 6 to 12 hours of light and dark condition respectively, and maintaining those cultures at 28 degrees Celsius. Again, lower light intensity has to be enabled for the pollen culture. So once the pollen cultures have been maintained or incubated for different time period, or about 20, 30, 40, 45, 50, 60 days, then there is a chance of formation of the callus, and those callus may directly, the callus may form, or directly the embryos can be formed. So if the embryos have been directly formed from the pollen grains, these embryos can be transferred onto a new freshly prepared nutrient medium, and those may directly form the haploid plant. Or if you have in the callus, you need to maturate those callus to form the embryos. Those embryos again they have to cross all the three stages. Okay, na? So to form a complete plant from an embryo, the three stages, uh, globular, heart, and a torpedo stage that has to cross. And once the callus have been forming the embryo, that embryo has to transfer again to a fresh, newly prepared nutrient medium, which is usually containing the plant growth regulators, which are rich in cytokinins, and low concentration of the auxins has to be prepared, and that can be uh, that can produce the haploid plants. So these haploid plants can be diploid, nothing but the cells of the particular plant which have been produced from the pollen grain. Each and every cell contains haploid set of chromosomes. Okay, now that particular haploid set of chromosomes can be doubled. It can be dihaploid or diploid, can be made by treating those haploid, what we call the cells into colchicine. Through colchicine, we can double the chromosome and we can make them as a diploid plant. Okay, now this is what the anther culture and the pollen culture. The comparison. So what are the applications or advantages or limitations or disadvantages of anther culture to the pollen culture? Okay, now coming to the anther culture, what is the application of the or advantage of having the anther culture and the pollen culture? So when it comes to the anther culture, this anther culture is very easy because we need to go for selection of a immature bud. You just need to surface sterilize them, and once the surface sterilization has been done, those flower bud has to be brought to the laminar airflow. Once the laminar airflow has been set right for the anther culture, you can go for aseptically cut the anther and identify the stage and put it on a nutrient medium. So it's very easy, quick, and very practicable. So when it comes to the pollen culture, it's quite uh, uh, tedious. Why? Because the here we need to go for removal of all the anther tissue debris. Only a pure pollen grains has to be selected. Okay. So earlier anther, which containing anther walls, anther tissues, pollen grains, everything should be there, will be there, and we are going to inoculate it on a nutrient medium. So here, once after identifying the flower bud, surface sterilization of the flower bud, you are supposed to select the anther. That particular anther has to be removed in such a way that there should not be any anther wall debris or anther tissue debris in the suspension medium. It should contain only the pollen. So after the once the pollen has been selected, there you need to go for microscopically observe the larger viable pollens, and those needs to be selected, and that has to be inoculated in a nutrient medium. So that this what one demerit of the pollen culture. But what are the limitations of the anther culture is the chances of, because of anther walls which usually will be in diploid condition, only the pollen mother cell which have undergone the meiotic division, that's what we call them as microspore mother cell, after undergoing the meiotic division, which is going to form the tetrad microspores, and these microspores usually contains a haploid set of chromosomes. And when we inoculate the anthers on the nutrient medium, because of the presence of anther tissue, other wall tissues or tapetum, epidermis, endodermis, or sometimes microspore mother cell directly, all these together may develop to form a diploid plant. Along with the pollen derived haploid plants, there are chances of diploid plant production too. So the culture which have been produced from the anther contains a varied ploidy. Nothing but the population. Some plants will be having a haploid condition, some plants will be having the diploid condition. So the ploidy of that particular plants which have been produced through in vitro culture using the anthers may have haploid plants as well as the diploid plants. Okay, this is what one big limitation with regard to the anther culture. Where in case of the pollen culture, because of undesirable effects or because of the because of varied ploidy population, we can avoid the anther wall. So by inoculating only the pollen culture, we can go for only the production of an haploid plant. Though the pollen culture is tedious, but the production of the haploid plants is very precise if the person is skillful. So because there is no disturbances occurred by any anther tissues. Only the pollens are going to be selected. The undesirable effects during the course of results can be avoided because the pollen culture doesn't contain any of the anther tissues. Okay, na? And during the course of the in any course of the plant tissue culture, since we are working with the pollen alone, single pollens we are taking, nothing but we are not looking at what happens, the anther tissues, walls, debris, all are not involved during the pollen culture. So we can easily regulate the developmental stages of the pollen because only single pollen will be there, and we can each and every stages, each and every developmental movement we can observe, we can identify, we can study, and we can regulate. But where in case of anther culture, there is a quite difficulty because the plants are birds which are going to produce from the anthers. We cannot regulate from which cells or from which part or from which area the plants in vitro are producing, because we cannot control whether the plants which are producing from the pollen mother cell or pollen grains or microspores or anther walls, or it may be from epidermis or from tapetum or from endodermis. We cannot regulate that, we cannot control, we cannot identify, we cannot easily find out which are responsible for the production of a plant. And even after the obtaining the result also, even after obtaining the plants also, we are not sure that whether the plants which have been obtained from the anthers are haploid or diploid. That has to be, again we have to take it for studying the mitosis. Once after mitosis study has been conducted, there you can go for, there you can identify whether they are haploid or diploid. Where in case of pollen culture, the regulation can be better because we are working with single cells or only pollen cells. Next, once the microspores or a pollen which have been taken, since we can easily regulate them, and we can once because of the pollen grain which are having a weaker cell wall, we can easily genetically modify them. We can go for conjugation, we can go for genetic transformation, nothing but we can easily introduce foreign gene, because these microspore are a pollen grain which are single cell, nothing but they are single or uniform cells which can be easily regulated, and they are having a very weak what we call cell wall, and we can easily make sure that nothing but the microspores can be easily taken for the genetic modification by using genetic engineering techniques. And the thing is that the yield of the haploids will be more with the pollen culture, because anther culture chances of production of varied haploid is greater in anther culture, because we are using only the pollens as the source of the starting material. Then the chances of yield are relatively higher than any type of the androecium derived part which is being taken for the production of male haploid plants. Okay, na? So these are the advantages or disadvantages or applications or limitations or merits and the demerits which have been between the anther culture and the pollen culture. To show you the flow chart how we can go for the culturing of anther and the pollen culture. And from my right side you could see the anther culture, where we can go for the flower buds. Immature flower buds has to be taken, and the flower has to be the selected and surface sterilized, and from there the anther has to be selected. Once the anther has been isolated, those has to be inoculated on a nutrient medium. So initially what happens, anther may form, or anthers proliferates to form the callus, and these callus usually, because they have been produced from the anthers, it has been observed, it has been noted that these are can be haploid callus. So once the callus have been formed, it may directly form the shoots or it may form the haploid embryos. These embryos, by treating with the colchicine, we can go for production of diploid plant, nothing but instead of producing an haploid plant, if you treat those with colchicine, as I already told you that these colchicine are the chief agents of conducting or happening, or which way enables the chromosomal aberration in such a way that we are going to see exactly double of the chromosome number. So these embryos can be treated with the colchicine to produce the diploid plants, or directly from the shoot we can go for production of a haploid plant. If you want to retain only the haploid plant, you can retain it. For future study, if you want to double the chromosome, and we can go for treating the colchicine, and we can get the diploid doubled haploid plant or a dihaploid plants or a diploid plant in a homozygous plant. Or during the pollen culture, you can go for the pollen isolation. Nothing but the anther has to be removed, only the pollen grains has to be collected, and those pollen grains has to be filtered, washed and collected. And once the filtered or washed, viable large pollen grains have been isolated, those has to be transferred on a liquid or a solid nutrient medium. When the pollen culture has been ready, they may form the multinucleate pollen, or nothing but there we can see the pollen grains are proliferating, nothing but the number of pollens are doubling. Then for we are going to see the formation of embryos, globular heart. This is just see the globular stage and the heart or a pollen embryo. This finally results in the formation of a haploid plant. Again, this haploid plant, you can stop the work here itself, or you can, oh my god, or you can go for treating your particular plant with the colchicine to get double haploid or dihaploid or diploid plant. So this is what with regard to the anther and the pollen culture. Now coming to the okay, we are at the last bit of the anther culture. Now coming to the what are the factors which are responsible or which can affect the androgenesis. So it may be with regard to the anther culture, it may be with regard to the pollen culture. In generally, what are the factors which can affect the androgenesis when you perform both? It may be the anther culture or the pollen culture. Excuse me. Now coming to the first factor which affecting the androgenesis is a genotype of a donor plant. Nothing but what do you mean by genotype of a donor plant? Nothing but the plant, the genotypic condition of the donor plant should be very viable. Nothing but it should not be forming non-viable or dormant pollen grains. Okay, now so donor plant, the genotype of a particular plant should be in such a way that it should produce all the pollen grains, all the anthers, all the stamens should be fertile, should be viable, should be greater active. Okay, now, so if there are any chances of infertile, non-viable, dormant pollen grains, then what happens? The androgenesis cannot be conducted successfully. Okay, now the person who is working with the androgenesis using what we call non-viable pollen grain, then non-viable anther, then the chances of obtaining the haploid plant is quite difficult. Okay, now so genotype for donor plant should be a greater importance during the androgenesis. Next thing is that, as I already told you, that you need to identify at what stage the anther has been selected, because appropriate stage of the microspore can give a better successful results. Okay, now, so the stage of microspore or a pollen should be between the tetrad to binucleate. Okay, now it should not be below or the early stage of the tetrad or post binucleate stage. The microspore or a pollen should be between a pollen tetrad condition to the each microspore or a pollen comprising the binucleate stage. Okay, now this pollen between the tetrad and the binucleate stage, this staged pollen grain which will give a greater results than any other stage or early tetrad stage or a post binucleate stage. Then after the third factor which is affecting the androgenesis is the physiological status of a donor plant. Nothing but the plant should be grown in a natural, beautiful, adjustable climatic condition. It should not be under any of the natural stress, not like abiotic stress or abiotic stress. Okay, now if the plant which is growing very robustly, healthy in a very comfortable situation, then the physiological condition of the plant is very high level. Okay, now at that condition, the results can be very pretty. So if the conditions or the physiological condition of the donor plant or a pollen donating or anther donating plant is under severe stress or a mild stress, then the chance of production of a anther and pollen is can be there will be a variation in the production of healthy pollen and anthers. Okay, na? So it should be grown at a appropriate light, appropriate what we call them as natural condition, water, humidity, the temperature, everything should be comfortable for the plant. So when the physiological condition is very good, the results are going to be very pretty. The next is anther pre-treatment. Sometimes we can to enhance or to make the results little better or better, we can pre-treat before the inoculation of the anthers with chemicals, or we can influence some of the different varied temperature to get the better results from the anther cultures. So usually treating the anthers with ethyl phosphonic acid can enhance the formation and which is going to reduce the period which is taking for production of the haploid plants. Treating the anthers prior to the inoculation with ethyl phosphonic acid enhances the chances or number of haploid plants and also decreases the time required for the production of an haploid. Okay, na? Or it is this is the ethyl phosphonic acid may enable the anther can increase the shelf life of anther for longer period, or we can different varied the different other pre-treatments can be done, such as that influence of the temperature. Nothing but we can store those anthers at low temperatures for 2, 3, or 4 days, depending upon the type of a pollen or anther you have taken. Okay, now so according to the retaining condition or the holding temperature holding capacity of the anther, usually 3 to 6 degrees Celsius, low temperature can be used for storing of the anthers for 2 to 3 or 4 days. These low temperature treated anthers which will give greater results. Okay, now that this has been already practiced. Okay, now so influence of low temperature in barley and in maize at 3 to 6 degrees Celsius for 3 days has given a greater results direct instead of directly inoculating the anthers. Now coming to the fifth important factor which is affecting the androgenesis is the light. Okay, now what happens? The production of haploid can be better in light condition. Nothing but there should be increased light. That's what we have mentioned it as 12 to 18 hours of light. Once the haploid plants have started forming the buds, then they require high light. During the study, during the previous slide, I had informed you that the lower light intensity is required for the formation when the pollen or anther have been inoculated. Once the bud formation or once the embryo has started maturating at a cotyledonary stage, there the better light condition is very good for the production of an haploid. Haploid production is very greater in the light condition. Okay, na? And next is the effect of culture medium. So usually the Murashige Skog medium, B5 medium, Mist medium, Linsmer Skog medium, Gamborg medium, any different medium can be used according to the trial and errors. Okay, na? And usually this all the nutrient media must contain the macro and the micronutrients. Apart from that, if for obtaining the plants, haploid plants, either from the anther or a pollen, they requires some specific plant growth regulators. This particular plant growth regulator are a medium augmented with rich or increased concentration of a cytokinin followed by low concentration of the auxin. So when you are using the combination of the cytokinin and auxin in the nutrient medium for the anther or pollen culture, they requires high concentration of a cytokinin and the low concentration of the auxins. So if you increase the auxin and decrease the cytokinin, there is a chance of only the callus formation. Okay, or alone you can go for utilizing only the cytokinins for the haploid production. And specifically we can even add the glutathione and ascorbic acid. So these again further acts as an add-on benefit. Sometimes you can even add the coconut water. So different type of add-on nutrients can be added for obtaining the haploid plants. So these are the factors which are responsible or affect the androgenesis.